Now, this is the overall or the overview. As you can see, you would see that the red, which is adenine, pairs with the yellow, which is thymine. Everywhere you would see that red is paired with yellow because it's adenine and thymine. Always. The green with the blue, as you can see, always guanine with cytosine. That must never change. Now, if you have a look at this, let's just go back to this diagram. Now here, you can see TA, TA, CG, CG, GC, GC, AT, AT, TA, TA, AT, AT. Just like that. This is proof to us that these two DNA molecules are identical to each other. Right? Because you can see even the nitrogenous bases, the sequence of nitrogenous bases and the pairing of nitrogenous bases is exactly the same. Okay. All right. Now let's move on. Okay. Now let's move on to our first activity. Let's have a look at activity 1. Like I said earlier, read this information. Read it twice if you have to. Make sure that you read. Now, remember, in your examination, you are given 5 minutes reading time. Don't just sit there while you're given this reading time. Go through your paper, read all these things so that you don't waste a lot of your time reading when the exam actually starts. Go through everything, read everything, or as much as you can so that you save your time when the exam starts. It says read the text below and answer questions. Read everything. Wherever you see words, read them. Right? It says here, Watson and Crick were only two of many scientists working on figuring out the structure of DNA. In the previous lesson, we looked at the history of DNA or the history of the discovery of DNA. So you know who we're talking about when we're talking about Watson and Crick. All right. Okay. They determined that the structure of DNA was a double helix polymer or a spiral of 2 DNA strands, each containing a long chain of monomer nucleotides wound around each other. Monomers, we're talking about the building blocks thing. Okay, according to their findings, DNA replicated itself. DNA made a copy of itself. They furthermore discovered that DNA is the basis for heredity. We know that one of the functions of DNA is the inheritance of hereditary characteristics from parent to offspring. It contains the patterns for constructing proteins in the body, including the various enzymes. We know that it carries the code for certain characteristics, including protein synthesis. A new understanding of heredity and hereditary disease was possible once it was determined that DNA consists of 2 chains twisted around each other, or double helices of alternating phosphates and sugar groups, and that the 2 chains are held together by bonds between pairs of nitrogenous bases, adenine, cytosine, thymine, and guanine. All right, now here we have question 1.4.1 up to 1.4.3. I'm just going to give you about 10 minutes. Remember, 1 minute, 1 mark. You should actually be getting 9 minutes, but 10 minutes will be fine. Go through these questions and then we will discuss them. Okay, so we're just going to quickly talk about fingerprints and why they are different from DNA profiles, right? So when we look at our fingerprints, we know that we all have our unique fingerprints, right? However, identical twins have the same fingerprints. They also have the same DNA. So we cannot change that about twins. However, everyone else have different fingerprints, but these fingerprints can have some problems. Now, these problems are what we would prefer using DNA profiles over fingerprints. With fingerprints, there can be things that can be done to change your fingerprints or To hide your fingerprints, if you know that you will be committing a crime, you can wear gloves. You can change the fingerprints through surgery to make sure that your fingertips are not identifiable wherever you have touched, which is why, like I'm saying, we can rely more on DNA profiles than the fingerprints, right? So these ones, they are found in every cell of your body. Remember, all the cells of your body are identical, right? Because they were produced by mitosis, right? Remember, mitosis produces cells that are the same. So the DNA is identical throughout the body, and it cannot be altered. I mean, you cannot change every single part of your body just to change the DNA. So DNA cannot be changed, right? So we find that DNA is useful in identifying individuals. Actually, we can identify individuals in ways that would not be possible with fingerprints, right? Now, this DNA profile, as we can see here, these are the black bars that are left behind an X-ray film when we extract DNA from an individual, and then that DNA is going to undergo some biochemical process. So here, these bands that we can see, those are representing our DNA, right? So based on your DNA, an X-ray film will form these bars. Now, these bars will be unique to each individual because we are not the same, right? Okay. Now, let's look at the interpretation of the DNA profile, right? So here we have a victim, we have evidence found from a crime scene, and we have 3 suspects. So what will happen here is that the DNA profiling process is going to be done by identifying an individual by comparing their DNA, these individuals' DNA, to another DNA that is known. So we know that the criminal was at the crime scene, right? And we are going to compare these suspects to the genetic material that was found at the crime scene to then say, okay, this belongs to the individual that matches the DNA from the crime scene, right? So you are going to compare the bands on a DNA profile of an individual to those of the DNA profile that is found on the crime scene or on another victim. You can sometimes find in rape victims that the DNA profiles of the suspect can also be found in the victim. Now, I said earlier, make sure you use a ruler when you compare the DNA profiles. So you're going to use your ruler. You're going to place your ruler here so that you can compare. Now we can see that suspect 1 already matches. Suspect 2. Suspect 1 matches somewhere, but not completely, like that. So if you are using a ruler, it will be very accurate. You will be able to see that which suspect matches the most. Now, as you're using your ruler, you can see that this one matches with suspect 2. So you're going to put a star there just to identify that this one matches. And then here it matches with 1 and 2. You put stars like that, so that at the end you are able to count which suspect has more bands matching. So as we can see, suspect 2 matches with all of the bands from the crime scene. Then that places him in the crime scene. What do we use these DNA profiles for? Like we've seen now, to identify criminals, right? We use it as biological evidence in order to identify criminals in a crime scene. We can also use this to diagnose genetic disorders. We can diagnose disorders with it. We can also use it to develop cures. So from our knowledge of the disorders, we can then use that knowledge to develop certain cures for different disorders. Biological evidence. What do we mean by biological evidence? Identifying criminals in crime scenes. It can be whereby there was a fight and someone was injured or killed. We can find out who else was at the crime scene, right? A rape case, if a victim says, this is the person who raped me, their DNA profile can be extracted and compared to the DNA profile that can be found in the body of the victim, and then that is how we can then say, okay, this is indeed true, right? Identification of relatives. With identification of relatives, we can look at it in many ways, because here it can be paternity testing, it can be identification of siblings, it can be identification of dead bodies as well, right? So An individual can be burnt beyond recognition, but if you suspect that this person belongs to a certain family, their family members can come, provide their DNA profiles, and in that way, we can compare whether the DNA profile of the living relative matches with the deceased, then we can identify them. If it does not match, then it is not the family member. But however, if it matches, then we can say that this is actually the person who belongs to that family. Okay, as I've said, the diagnosis of inherited disorders. So DNA profiling can be used to diagnose disorders such as hemophilia and sickle cell anemia, right? So the diseases may be diagnosed in newborns as well as in unborn children. It can be a bit complicated when we're looking at identification in unborn children, but it is still possible. Developing cures for the inherited disorders. So for the inherited disorders that we've identified, looking at the DNA profiles and being able to see the causes and the problems around it, we can then use that knowledge to try and develop some cures. Right, the researchers can use DNA profiling to find patterns in large groups of people when with or without the disorder. Okay, so that understanding is a necessary step in finding the cure. Right, biological evidence. So it can be in forensic science to find criminals. Now, like I said, we can find criminals using their fingerprints, but that can be changed. Now, if for some reason the criminals have left their DNA behind, we can use that DNA to identify them. Right, now, this does not tell us whether the individual has committed a crime or not. It simply tells us that the person was present at the place of the crime. Now, from there, knowing that the person was at the place of the crime, we can be able to take steps going forward. But it does not say this person has committed the crime. Okay. Right. So we can use things such as your blood, your semen, your hair, your saliva, skin cells. These can all be used as sources of your DNA, right? So that is where you can supply your biological evidence from, right? So this will then be used to create the DNA profile, right? The DNA will be extracted, used to create that profile, and now that profile is what we are going to see in the form of those black bands, which we are going to compare. Identification of relatives, right? Firstly, determining the father of a child, like I said, paternity testing, right? Tracing siblings, we can suspect that the two individuals are siblings. Some of their DNA profiles are going to match with each other, and therefore we can then say that these are siblings, right? Because they got their genetic material from the same parents, remember. Okay, identifying people who have died and could not be identified. So that is also another thing that we can use it for. Now, here we have two DNA profiles. We have the DNA profile whereby it can be used for paternity testing, and we have a DNA profile whereby it can be used for identifying a criminal. These two are not necessarily assessed the same way or interpreted the same way. Let's start with the one at the crime scene. Right, so we have semen. It means that we are looking at the semen that was obtained from the victim, right? And we have the three suspects. Now we are going to compare these profiles with the profile of the victim and the profile of the semen and the suspects. Remember, the semen profile can also contain some DNA from the victim because it was obtained from the body of the victim, isn't it? So here we can see this first one matches with the suspect and the victim, right? So I've marked where it matches. Then the second one, it matches with suspect number 3. Already the odds are against suspect number 3, but we're just going to carry on so that we can make sure that the results are 100% correct. The third one, that light one, it matches the victim and suspect 3, as well as suspect 1, right? Okay, actually, if we are looking at this, it could possibly just match suspect 3. This is why I said make sure you bring a ruler so that you can place your ruler here and be able to see exactly where the line is placed. Okay, the next one matches suspect 3. This one again, suspect 3. Next one, suspect 3. That big one, suspect 2 and 3. The last one, suspect 3 as well. Now, from here, we can conclude that suspect 3 is the person who committed the crime. Now, when we are looking at paternity testing, we are going to focus on the mother and the child first. Why? Remember, this DNA profile of the child has 6 bands, as we can see here. It means that of these 6 bands, 3 bands are supposed to match the mother and 3 bands are supposed to match the father. We start with the mother so that we can identify the ones that match the mother. Why do we start with the mother? We know that she's the mother. So definitely 3 bands are going to match her. The remaining must match the father. All of them. If only 2 matches all these men, it means that they are not the fathers of the child. But if there's a man here who has 3 bands matching with the remaining 3 of the child, then that is the father. Let's start. First band matches the mother. It means that when we match the father now, we are not going to look at the first band because we have already finished with the first band. I'm going to put a tick here to show that we are done with the first band. Second band, it doesn't match the mother. We leave it out for now. Third band, it matches the mother, right? The fourth band, it doesn't match the mother. It means the fourth band must match the father. Number 5, it matches the mother. Number 6 does not match the mother. Now, how many bands of the child are matching with the mother now? 3 bands. Like I said, the next 3 must match the father. We're going to compare number 2, number 4, and number 6 with the possible fathers. Number 2 matches with male number D. Now, can you see that it could possibly match with B, but it's slightly lower, right? So we tick. Let's just put a dot there, right? And then number 4 matches with A, B, and C. It doesn't match D completely. We are ruling out D, but we can also rule out A and C because the first one did not match. And then number 6 matches with B. So it's definitely B. Unfortunately, here it wasn't placed as correct as it should, but it means that male B is now the father of this child. Okay, but we can have more profiles just to confirm that, because as we can see, there is a bit of a doubt there, but to make sure we can check more. Remember, this is just a portion of the DNA that we have obtained from these individuals. The DNA can be actually much bigger than this. Right, let's have a look at the next activity here. Activity 2.3. Now, read this information. Before you get to your questions, read this information, understand what this information says, then you can move on to your questions. Mrs. Jill had baked a birthday cake for her sister, Anna Marie, and kept it in a tin on the kitchen table. When she returned from shopping, all that was left in the cake tin was a few crumbs and a smudge of blood. Now that is our evidence, where the thief had snagged his or her finger on the sharp edge of the tin. Study the DNA profile of her 4 children given below. So we have the blood stain from the tin with Bob, Sue, John, and Lisa. Those are Mrs. Jill's children. So we want to find out which of the children have stolen the cake. Now, I'm going to give you 8 minutes to go through these questions. Then we are going to go through them together. All right, now let's look at the questions together. The first question is 2.3.1. It says, define the scientific term DNA profile. What is a DNA profile? Remember, when we talk about a DNA profile, we will be talking about the black bands or the black bars that are found on an X-ray from the DNA that was provided by an individual. So you can answer it two ways. It is an arrangement of the black bars, lines, or stripes representing the DNA fragments of a person, right? Or you can say the pattern of bars that indicate the sequence of bases on a DNA strand. Okay, number 2. Which one of Mrs. Jill's children is most probably the suspect from the description given above? Give a reason for your answer. Now let's go back. We are going to compare these profiles with the blood stain from the tin. Okay, now this can be a bit difficult. Why? These are siblings. It means that most of their DNA profiles are the same. But remember, they are not twins, therefore identical twins, therefore they're not going to be exactly the same, but mostly the same. The first one matches everyone, right? The second one, Bob, John, and Lisa, right? As you can see, we're finding a lot that are similar here. And then Sue and John, we can already see that John is the one that has a bit more matching than the others. Next one, Bob and John. Again, Bob, Sue, John. Next one, Bob, John, Lisa. Next one, Bob, Sue, all of them. Next one, only John. The next one, John and Bob. So as we can see, all of them are matching with John. So John is the one that has taken the cake, actually. So the reason here would be that all the bars on John's DNA profile are matching with all the bars from the blood stains that were obtained on the tin. The DNA profile of John matches with the DNA profile of the blood sample collected from the edge of the tin. Describe how DNA profiling can be used for paternity testing. So here, remember, for paternity testing, the dark bands that are seen on the DNA profile of the child is a combination of the father and the mother. It means that the dark bands of the child's DNA profile must match those of the parents. And then the next one, do you think that saliva could also be used as a sample material in this type of investigation? Motivate your answer. Yes, it can be used. Remember, saliva also contains DNA. Right, so from the lining of the mouth we have DNA. Therefore, that DNA from the saliva can be used as DNA evidence. Okay, activity 3. This is our last activity for today. It says the graph below represents the result of an investigation which compares the conviction number of people found guilty of the conviction of criminals with the use of DNA evidence and without the use of DNA evidence from 1989 to 2003. There we have the graph. The first line, that is with DNA evidence. The second one is without DNA evidence. We can also see that without DNA evidence, it's lower than with DNA evidence. And as the years progresses, the technology has improved. So the number of convictions have increased much higher than without the DNA evidence. All right, we have a 9-mark question here. 10 minutes, go through the question. Make sure that you attempt all of these questions. Read the questions, read the information, interpret that graph, and then we'll come back and go through the questions together. Okay, so it says here, formulate a hypothesis for this investigation. So we've already discussed how to formulate a hypothesis. Remember, by looking at the graph, you can already see what the hypothesis would be in this case. You can see that with the use of DNA evidence has increased the number of convictions, right? However, you can say that the number of convictions have increased without DNA evidence. You don't have to be exactly correct. It is a hypothesis after all. But let's have a look at this one. It says, using or not using DNA evidence increases or decreases the number of people found guilty. So you can say using DNA evidence increases the number of people found guilty. Or you can say using DNA evidence decreases the number. It's up to you, right? Or you can say not using DNA evidence increases or not using DNA evidence decreases. It is up to you. You would still get your 2 marks. Or you can say using or not using DNA evidence has no effect on finding people guilty. So that is your hypothesis. Using DNA evidence is your independent variable because the use of DNA evidence has an effect on the number of convictions. So the number of convictions is our dependent variable. 3.2 says state the dependent variable. We've already mentioned that the dependent variable would be the number of people found guilty, number of convictions. 3.3, name how many more successful convictions per 1,000 arrests were made in 2003 with the use of DNA evidence. Now, here, mathematics. Make sure that you bring your calculator so that you can calculate the results that you're going to get. Now, let's go back to the graph. Okay, now on the graph, we can see that with evidence, we have 44 here, right? Just below 45. And then without evidence, we have around 20, right? So now it means that we're going to look at the difference between what we have when there is DNA evidence and what we have when there is no DNA evidence. So, Rosalani? All right, so with DNA evidence, we have 44 convictions, and then without DNA evidence, we have 25 convictions as seen on the graph. So it means that there is a difference of 19 convictions. That is a big number. So there's 19 more convictions as compared to when there is no DNA evidence. Okay, a DNA database is a collection of the DNA profiles of all citizens of a country. Explain how you would use the information in the graph to convince the government to create a database, a DNA database. From the graph, you can see that the difference here, as we have seen, 19 more convictions as compared to when there is no DNA evidence. So you can simply explain that from 1989 until 2003, there has always been a high number of convictions when there is DNA evidence as compared to when there is no DNA evidence. Now, that is the biggest reason why the government should then consider using this DNA evidence as something that they can use to convict criminals. So more criminals are found guilty when DNA evidence is included. DNA found at crime scenes can be compared to the database. So because now we have a database for everyone, we can simply compare to the database and quickly find the criminals without even having to try and ask those criminals to cooperate and provide their profiles. All right. And then this will then make it easier and faster to identify these crimes. Okay. Now let's just look at the summary of what we did today. DNA replication. Double strand unwinds. As it unwinds, the weak hydrogen bonds are going to break and the double helix is going to unzip. As it unzips, we are now going to see that the two strands act as templates so that we can have the formation of the new strands using complementary base pairs, right? Now, the building blocks, which are the nucleotides from the nucleoplasm, are going to come and join to their complementary bases. This will now result in two genetically identical DNA strands. One would have one old strand and one new strand. And remember, this process is controlled by enzymes. That is what you would look at when you're looking at DNA profiling questions. And then here, mainly, what is the use of this? Parentage testing, paternity testing, medical diagnosis, diagnosing genetic disorder, and trying to find the cures for those. It can also be used in agriculture and the food industry, and mainly in forensic science, as we have discussed, identifying of criminals, right? So look at the uses, look at how to interpret this DNA profiles better, like I said, use your ruler and also understand how to identify the bands as you are comparing the DNA profile of the mother, the child, and the possible fathers during paternity testing. Now, please make sure that you bring these things that we have mentioned here in your examination room. Make sure that you have them. Once you have these, you will be sure that whatever question you are asked, you are able to answer that. You cannot miss marks because you did not bring your calculator. You cannot miss drawing a graph because you did not bring your pencil and eraser. Bring an extra pen as well, just in case the one that you have stops working. Your protector and compass, make sure you bring those in case you are expected to draw a circle or a pie chart. Your ruler, make sure you have your ruler so that you can also use that for your graphs, your tables, and as I've said, in your DNA profiling questions. Now, in the next lesson, we are now going to be looking at protein synthesis. We're going to look at what happens in the nucleus during transcription, what happens in the cytoplasm during translation. And we are also going to look at the functions or the role of mRNA and tRNA during this process. All right, we have come to the end of our lesson for today. Thank you for watching. I will see you next time. Goodbye.